Now, I am a selfish person. I did not become a scientist because I wanted to make the world a better place. I became a scientist because I wanted to do things that I really enjoy, analyze data. In school, I did not like arts. Like, I didn't really know how to make colors match or how to do anything that other people actually like to look at. I don't appreciate art. Well, I've been, of course, to the National Gallery because I was in London and it was, raining. They have a free entrance in the cafe. I did not even stop at the most famous paintings to take a selfie. And I think that really shows something about my lack of interest towards art. Right now, I am analyzing biological data to improve healthcare. So I guess I am making the world a better place, theoretically. Because in practice, what I really do from day to day is to create beautiful plots for people to look at. Let me exemplify that to you. Let me share a screen. I will show how this will look like in my work. I hope this works. And I hope you are seeing my screen. Let me see if I can present that. I expect you're seeing my screen. If you're not, shout out loud.
It's fine, we can all see it.
Thank you. So an example of how you can present your results when you get the science, that's how it looks you in the face. What you could do to tune up your plot, well, you could just, instead of the boring circles, have some variety, add triangles, maybe squares, maybe crosses, just anything else besides the boring old dots and circles. Another interesting thing you could do is add colors, because that will really lighten it up. The science comes alive, with this deep pink and dark gray and forest green. That's a great combination. More things you could do, you could add lines. And not just the regular lines, but dotted lines or dashed lines, something. Just add more gadgets to the plot. It will look actually, to be honest with you, this is much better science. And I have to be honest, that's unfortunately true. So I spend not minutes, not hours, I spend days perfecting my plots, because when I show up to a meeting showing that kind of plot, people want to discuss science with me. But if I should show up with black and white plot, or worse, default colors in Excel, I assumedly have no personality, no motivation for research, and definitely no scientific credibility. What I do now is that I actually analyze single-cell RNA sequencing results. Single-cell RNA sequencing, this is like the coolest method ever for now in biology. The method is sort of like, imagine if you get a new toy. What you want to do with that toy is you want to re-experience everything you've seen. You want to take that toy to the world tour with you to show everything you have already seen. And that's what we do with single-cell RNA sequencing. We are rediscovering the biology of an entire human being with that new method. And my part is to rediscover the entire biology of the human lungs. So far, I have found 56 new cell types. 56, that's a great number, if I say so myself. That's really something, good about the science. Unfortunately, I have spent months for all of those 56 cell types to choose a color of their own. There are so many colors to choose from if you only know. There is green, green 1, green 2, aquamarine, deep medium sea green, cobalt green, lime green, spring green, aquamarine 2, aquamarine 3, aquamarine 2. It's so many shades of green. I did not know they exist. If I had studied arts, maybe I would have known. But no, I went for science, the serious stuff. Yellows, yellow 1, yellow 2, yellow 3, goldenrod, gold, banana, goldenrod 2, light goldenrod, dark goldenrod. I could go on and on. But it's not just enough to pick 56 random colors of that. You have to choose the colors that are pleasing to look at, to the eye. 56 completely distinguishable colors. Of course, 56 distinguishable colors. Ideally similar between the cell types. And of course, if you can, colorblind compatible, red-green colorblind compatible. And that makes my life really difficult. But there is a bit of a silver lining. With this new method, with this single-cell RNA sequencing method, there is a potential to discover new cell types. Now, that is a great finding as a scientist. That's why I'm doing science, to find something new, to discover new things. It could also raise impact of my work. It could define my future career. And in this lung, in these human lungs, I discovered a new cell type just last week. And that's essentially all that I can think about, is that I had to choose another color. Now, I wish I could end on a more positive note, but unfortunately, things just go downhill from here. You see, single-cell RNA sequencing, that is so 2019, maybe 2020. The future is really for the spatial methods. We have Visium chips, spatial transcriptomics, Mirfish, Seqfish, all that kind of things. And one thing these methods have in common is that they're all essentially just art. So maybe next year, I'm really not looking forward to the time when I will become more of an artist than a scientist. And that's all from me. Thank you very much.